Now we pass over to the second speaker, Professor Bruno Lina from Lyon University. Professor Lina, the floor is yours.
Thank you very much, and thank you for this invitation to share with you some of the information. I'm sorry, I'm not sure I'm going to provide any very good data. One of the things I will provide to you is what we understand in terms of knowledge about the virus and how the virus is moving, or whether it is moving or not moving actually. So I just put my slides on the screen. Sorry about that. Should start from the beginning. So I want to go back to the different stage the virus went through during its emergence, and what is the evolution that we have seen in terms of evolution of the virological genome, and whether these evolution have had any impact in terms of transmission. So the virus, you have to remember from the very start, that this virus is a respiratory virus, obviously, but it is from the beginning very much transmissible. And when you look at these viruses, as you know, the coronaviruses, they have all the same shape. They look like the one I can show you here. It's a round virus with these little dots here around the envelope of the virus, which is making this virus called the coronavirus, because it's like a crown that you have around the virus. And this virus actually, when we grow this on epithelial cells, human epithelial cells, we could see that it was hugely well replicating, and the system was very efficient. So this virus was really fully adapted to humans. But we also knew that it was a new virus coming somehow from a reservoir, and the most likely reservoir for coronavirus are bats. And you know that we have had two previous experiences of the introduction of viruses from bats, which is the SARS and the MERS. And these two viruses came from this reservoir, jumped into another species, which is the civet cat for SARS, and the dromedary for the camel, sorry, for the MERS. But even though these viruses went to another species and then subsequently went to the man, they were not fully adapted to humans, because the transmissibility of these viruses were not as good as what we have seen with the SARS-CoV-2, although there were some cases of transmission and some cases of very large transmission with the SARS. But still, we could get most easily control of these viruses by some very basic hygiene measure, things that doesn't happen with the SARS-CoV-2. And when we look at the very first genome that was available, that was a virus that has been collected in a case during the very early stage of the epidemic in the Wuhan wholesale market. Actually, this virus was really amongst the beta coronavirus, and it was very close to a bat virus that has been discovered a couple of months before. And the similarity between these two viruses was very high, more than 96%, as it is said here. And it was quite different as compared to other coronaviruses. So there was some kind of link, obvious link, between this virus and this bat virus and the emerging virus. And when we look at when we were looking for the animal that should have been involved into the transmission to humans, you know, because the bat is not frequently transmitting directly to human, we had the first idea that there might be the pangolin involved into the transmission, because in the Guangdong area, which is not exactly near Wuhan, but still it's in China, there has been a couple of these coronaviruses quite close to the SARS-CoV-2 that have been detected in these animals. But when you look at the phylogenetic tree of these viruses, actually you have the bat one here, which is very close to the human ones that are in blue. And you could see that the pangolin viruses actually are much more diverse than was the bat virus, which tells us, when you look carefully to this phylogeny of these viruses, that there might have been an introduction from this of this bad virus into the humans and the pangolin, but the connection between the pangolin virus and the human virus is really not obvious. So there might have been another animal involved into this transmission and the adaptation to humans, but it has not been identified as yet. And the thing is that when you look at the virus, when it was emerging, different viruses have been detected in China mostly, and we had the first French virus that was sequenced on the 8th of February. It was very close to the virus that was in the Wuhan market, these viruses were very similar. And what we did see from the very early stage is that these viruses that were identified were highly related, and the homology between all these viruses was very, very high. But still, at that time, we have the idea that this virus was highly transmissible. And this is due to the fact that actually, when you look at the bat virus, and you compare the bat virus with the previous SARS virus, at the spike protein, and looking at this at the protein sequence of the spike protein, which is involved in the attachment of the virus to the receptor being here in blue, you could see that there's huge difference between the SARS and the beta coronavirus that have been introduced into humans. So this is really a new virus, and this is really showed very well with the spike protein sequence. And when you look at the sequence, the protein sequence of the human SARS-CoV-2 virus, and you compare that with the sequence of the same regions, the spike of the precursor, the potential precursor that is in the bat, you can see that most of the structure of the protein is identical. There's only in the region which is supposed to interact with the human receptor, ACU2, that some changes have been observed. And these changes may not have been directly observed in the bat, because it would have prevented this virus to grow in the bat cells. So one of the possibilities is that through recombination with another virus, the rat TG13 bat virus somehow acquired the capability of binding very well to ACE, human receptor, and then subsequently was introduced in human, and then was able to be transmissible from human to human. And this allows the virus to replicate in the upper respiratory tract, which is the mandatory requirement for rapid transmission within humans. So we have not identified yet where and with which virus did this evolution of the rat TG13 virus so that it became a human virus with the capacity of being highly transmissible. Since the beginning of the epidemic, there has been an enormous amount of sequence data that has been generated, and this data is shared openly on the GEZ database, which is really an asset for the surveillance of the virus. And what we identified very early in March, and this was related to an evolution of the virus maybe at the end of February, is that we could identify 3 genogroups. And the 3 genogroups, the G here, S and V, were associated to specific substitutions that were occurred in different proteins. And the G protein here, the G group, actually is the one which is most interesting, because I will talk about this. And this virus is going to take over the other one. This is a substitution in the spike protein. And this substitution has somehow be overwhelming the other viruses, because when you look at what happens in less than 2 months' time, actually all the G virus became very much predominant, but it never completely displaced the other one. So the theory we had at that time is that maybe these viruses was having some kind of better fitness of the virus, and actually it was possible for them to have a higher transmissibility rate. But the data that we had very early on that was not really crystal clear, and there's still a work going on. But as if you look at the sequence data that is available yesterday, this is the picture where you could see that the G group have been subdivided in three subgroups, G, GR, and GH. And we still see some of these other genogroups. They don't completely disappear, they're still circulating, and we can detect them in different places in the world. So although we can say that this G group with the subgroups are really overwhelming the rest of the sequence that have been identified, these other subgroups remain and they have not disappeared yet. So when you look at what kind of substitution can be seen at the level of the protein which is interacting with the ACE receptor, there was this D614G substitution, which is close to the receptor binding, but not necessarily. And there's another group of substitution that has been recently identified from July and up to recently now, which occurred in the regions close to the receptor binding. And there's one very interesting, which is the S477 with the different possibility RY, RI, RN, where we say that these substitution may have an enhanced capacity, may induce an enhanced capacity to the binding of the receptor of the AC to AC receptor. This is an ongoing study, but if you look at the overall evolution of the viruses, and I take the example of what we have done in France in terms of genomic surveillance of the SARS-CoV-2, what we see is that within the sequence that have been sequenced from the early stage of the epidemic, saying from March until June, there's a very limited evolution of the virus. And this is due to the fact that we know that the coronaviruses have a proofreading system and it doesn't accept much of the sequence changes. There's a correction in the sequencing. So when we look at the molecular clock of the evolution of these viruses, we see that the change that occurs in these viruses is the maximum of 2 to 3 nucleotides per month worldwide, which means in a genome whose sequence is 30,000 viruses, a very limited evolution capacity. And when you look at the different SNP positions where this mutation can occur, there's also a very limited number of SNP positions that can be impacted, less than 500, which means that this genome is highly stable. And when we hear about mutations, substitution, risk of evolution, you have to remember that this is a coronavirus. This is not an influenza virus, and it is highly stable. So I just want to make an additional picture of what is the impact actually in Europe based on the ECDC data of this virus. And what we can see is that there's almost no country that can avoid having the circulation of this virus, where the virus is not evenly distributed over Europe, but still we can see that there are very large hotspots, and France is one of these. So in summary, and this is I have the acknowledgement slides after that, what we know is that the virus that emerged was actually perfectly fit when it emerged in December. And this was due to the fact that all the preliminary requirement for optimal dissemination in humans has been done when the virus was emerging. The genome is highly stable, as expected for the coronaviruses. However, we must keep in mind that there are possibilities for recombination with other coronaviruses, human ones, and deletion that may also impact the virus. The introduction of the D614G substitution in February that led to have a predominant virus, we are still working on the fact whether it is associated to an enhanced transmissibility. There's some very interesting work going on showing that maybe it was the case, but this is something that occurred in February. There's this recent emergence of the S477N substitution, and again, we are looking whether it has an increased affinity to the ACU2 receptor. And what I want to share with you also is that all what I can tell you now is based on the fact that there's a rapid sharing of the sequence data from the viruses. So if you don't do it, please share as much as possible the sequence data that you have. I will just acknowledge all the people that are working with me and providing the data that can be helpful for the discussion today. Thank you very much.
Thank you very much, Professor Lina, for this fantastic overview and this, as a virologist, I should say, very passionate work. And I am sure it will raise a lot of questions in relation with epidemiology and also in relation with protection and immunology. And